Okay, welcome to S.B Class. I'm going to talk about some topics that I saw, so we're going to start.
Inheritance is the process by which the characteristics of individuals are passed to their offspring. A gene is a unit of heredity that encodes information needed to produce proteins, cells, and entire organisms. Genes comprise segments of DNA ranging from a few hundred to many thousand of nucleotides in length. The location of a gene on a chromosome is called its locus, plural loci. Homologous chromosomes carry the same kinds of genes for the same characteristics. Genes for the same characteristic are found at the same loci on both homologous chromosomes. Genes for a characteristic found on homologous chromosomes may not be identical. Alternative versions of genes found at the same gene locus are called alleles.
Nucleic acid is nearly equal, wherein the cell chromatin usually folds into characteristic formations called chromosomes. Each chromosome contains a single double-strand piece of DNA along with the aforementioned packaging proteins. How much chromosomes are needed to create an organism? 22 of these pairs, called autosomes, look the same in both male and female. The 23rd part is the sex chromosome.
Heterozygous and homozygous. If both homologous chromosomes carry the same allele, gene form, at a given gene locus, the organism is homozygous at that locus. If two homologous chromosomes carry different alleles, at a given locus, the organism is heterozygous at that locus, a hybrid.
What are nucleotides? Nucleotides are organisms compounds that consist of three joint structures: a nitrogen base, a sugar, and a phosphate group. The most common nucleotides can be divided into two groups: primers and premixes. Based on the structures of the nitrogen base, the joint sugar is either ribose or deoxyribose. Nucleotides are the structures of units of RNA and DNA. They also serve as important cofactors in cellular signaling and metabolism. Their cofactors include CoA, flavin adenine dinucleotide, flavin mononucleotide, adenosine triphosphate, and nicotinamide adenine dinucleotide phosphate.
The Punnett square. Punnett square shows the genotypes two individuals can produce when crossed. To draw a square, write all possible allele combinations. One parent can contribute to its gametes across the top of a box, and all possible allele combinations from the other parent down the left side. The allele combinations along the top and sides become labels for rows and columns within the square. Complete the genotypes in the square by filling it in with the alleles from each parent, since all allele combinations are equally likely to occur. A Punnett square predicts the probability of a cross producing each genotype.
Phenotype. In this case, the plant that we used was the pea plant. So our phenotype is this one. In this part is the trait. You can see the seed shape, the seed color, the pod shape, the pod color, the flower color, and the flower location. The dominant form in the seed shape is smooth. In the seed color is green. In the pod shape is inflated. In the pod color is green. in the flower color is purple and the flower location at leaf junctions. And the recessive form in seed shape is wrinkle, in seed color is green, in pod shape is constructed, in pod color is yellow, in flower color is white, and in flower location is at tips of branches. Genotype. Our genotype is homozygous dominant, homozygous recessive, or heterozygous. This can be measured as fraction, as a ratio, as a percentage, or just the number. Don't forget that the homozygous dominant and the homozygous recessive are equal, and the heterozygous is the different. In this case, I'm gonna use the capital X like smooth, the lowercase x like wrinkle, the capital P as yellow, and the lowercase p as green. The F0 will be X capital, XX capital, XX lowercase, PP capital, and PP lowercase. In the F1, what we make is an exchange. So we make this with X capital with P, X capital with P lowercase, X lowercase with P capital, and X lowercase with P lowercase. So in the F2, we will have this by making this the F1 procedure. So with this, I can make our Punnett square, and in the Punnett square, I can know the phenotype and the genotype. With the F2 pattern, I must make the Punnett square. So that pattern, I use it in the first line and in this part. So how works our Punnett square? Okay, our Punnett square works like this. XP plus XP will be XXPP. XP plus X and lowercase p will be XXP lowercase p. So what we make is to combine those, and always the dominant will be at the first. If you look, the P is the final one, will never be in here. So can be in here because if it is X, but always at the final of the pattern. So with this, I can know our genotype and our phenotype. The homozygous dominant that we have here is this one, that is the only one in all capital letters. And the dominant recessive is this one because it's the only one that have lowercase. So with this, I can make predictions of generations.